And she also promised to get me some pictures today too, which I'm going to hold her to.
You can follow me.
I will.
Okay.
I'm all ready for it.
Mrs. Atkinson's class is also watching. She
Another
says hi.
stalk crinoid.
That's a cool Argus view.
Hi to them too.
Yeah, ooh, we're coming up on some structure.
It's because Argus is pretty much in the dirt.
Too bad we're all chock full of sediment cores.
Oh, we got another rock with some more.
Oh, this looks fun.
Yeah.
What is that giant
Is
thing?
that a
It's
giant
Whoa.
coral?
Oh,
It's
wow.
another black coral.
Whoa.
Like two stars.
I
Oh,
know.
oh, look, it's a battle between the squat lobster and a sea star trying to eat it.
Are those two squat lobsters or?
I think it's a
sea
squat
star?
lobster and a sea star.
Wait, where? What,
At the
what-
bottom?
Down towards the bottom.
Well,
Come
okay, we'll have
down.
to take a look.
It's gonna be tough to get over there.
Oh, okay, well,
Oh.
all right.
And this one seems to be missing some
It's
branches. Huh?
Yeah.
Some
I'm gonna
legs.
give you more slack by just facing away.
Okay.
You can
Thank you.
Can't
take a
zoom out.
gander at it.
yeah, it is missing some branches. Not really sure what caused that little anomaly right there in the center.
Oops.
More of these beautiful squat lobsters that we collected the other day.
No, it's two squat lobsters.
Two squats.
Double
Or-
squat.
Or are they squash, Aaron?
Just wait, you're gonna start accidentally saying that.
I know I am. There's a real danger there.
I'm gonna say it if I see someone land on one.
No. These blocks are kind of curious as to, how they actual formed. Did they actually roll downhill, or was everything eroded around them? Or I'm throwing a bone to the geologists out there. I don't know.
Chris, how varied is deep sea life from one area of the oceans to another?
It's a very good question, actually. There are
Whoopsies.
similar
Other way.
groups of animals, but the compositions can vary as the individual species vary. And what I mean by that is if we go to the Marianas
Oh, okay.
on the west
Thanks.
side of the Pacific and Hawaii on the east side of the
Turn right
or
more, please.
Central Pacific,
Sorry?
Turn right.
Turn right.
we can see at a certain
045.
depth
I'm
a
watching the tether in
bamboo-dominated
there.
community, but it's a
Okay.
bit different from Hawaii.
Because of me facing away right
And then
now.
we can
it's
go up
more
further and we
important.
can find a different
I can have it
community
face you
that's
again.
similar between the two, but they have different members.
That's the thing.
So
Try anyway.
there's similarities and then there's differences, if that makes sense.
This viewer is comparing Hawaii to the mid-Atlantic.
To the mid-Atlantic?
Yeah.
Yeah, you can keep
Okay.
going 045.
Well, there's going to be
Okay.
more differences there because you've got that big barrier of North and South America in the way, even though you get some flow around the tip. It's one of those big, huge chrysogorgids down there, too. But, yeah, it's a good question.
Oh, sorry.
And we saw some differences from seamount to seamount on this cruise, right?
Right. And actually, I was watching the Okeanos stepping stone
What's
expedition
the ground
from
called
home.
on Herc?
That was in the Atlantic a little while ago.
Gotcha,
And yeah, you can
okay.
see similar communities, but they have different members.
So Chris, as this is our last dive, what has been your favorite part?
Well, of course, the dive yesterday. Was it yesterday? I don't even know what day it was. That place was so astonishing. That was the best part. But also, I just really enjoyed everybody on the ship. It's just a wonderful company.
Oh, hi.
Everybody, and the weather was terrific. We were really blessed to have good weather for most of the cruise. I mean, there's a lot of good parts about this cruise that I like, but in terms of the biology, it was yesterday. That was just astonishing.
We have another question here about corals. Do all corals compete for space using digesting filaments? Would these corals attack one another?
Well, if you want to count zoanthids as corals, which they aren't really, they're a type of cnidarian related to corals, then they will attack other corals and overgrow them. But if you want to count like this type of community right here, they're not really doing that. Right here, there's enough space down there, I think. And
That's so cool.
This is beautiful.
Oh, that's gorgeous.
Zoom in, please, Aaron.
Take 100 photos.
Just even the
That's so cool.
even the backup view of the stand is just extraordinary.
Kill lasers?
What
Please.
is the name of this squirrel, Chris?
It's a Bathopathes, and that little squat lobster is a Europtychus new species. We've collected it now. We got a second specimen for the arthropod specialists now to help them
Trevor,
describe it.
can you kill the lasers? Please. Thank you.
Chris, it might be a little late, but do you want to try to hang out here?
If it's possible, just to,
Bridge hold position.
I want some
Or I can
more imagery of
hold
the
on. I can
lobster.
get it.
it's just so beautiful, just to get some shots of
Yep.
this too.
Yeah.
There's no real.
Wow. I don't see anything unusual here. It's just the beautiful composition is what it's got me.
Composition, that was the word I was looking for before. It's perfect composition.
And this beautiful
Hi, Justin.
composition seems to be on pretty large rocks.
Yeah, that's
Yeah,
correct.
definitely.
No sediment around those rocks.
And they're very stable. That's why they've been here for a very long time. And so they chose well when they were settling out. The little larvae did. They picked a good place. So this is what I think is what we're calling a telepathes. See if
The
the
little black
branches,
coral on the
yeah,
lower
the
left.
big tall black coral, it's branching on the right-hand side here. And most of the Bathopathes don't branch, so this is very confusing.
Zoom in, please, Aaron. Like a half zoom, maybe.
So is that a subspecies of the Bathopathes? It has that branch.
This, I think the black coral geneticists and morphologists are trying to figure this out right now. There may be a revision to what we think this thing is. If and that was a comment that Jeremy Horowitz made in the chat the other night. I hadn't noticed that. We came in and we were so high up that I didn't notice it was branching. It's simply gorgeous.
So about how many species of corals would you say are on this rock?
1, 2, 3, 4, 5, 6.
So funny.
Reach for the stars, buddy.
Yeah. That's a good motivational poster right there.
You know, 10 maybe or something, just on a little boulder like this.
I think that I'm going to caption that Sisyphus, eat your heart out.
All right, ready to go?
Yeah, thank you very much for
No
that.
worries.
That's beautiful.
Bridge nav, 20 meters, 140.
There is even a token sponge.
A token sponge.
Yeah, so Mother
Love it.
Deep Sea threw me a little bone.
Uh-huh.
And a viewer wants to know if there's any life under the sediment.
Yes.
And what kind of life would that be?
They
Besides
call
microbes?
well, they call it infauna, which means that it actually lives inside the
There's
sediment.
2 sponges, Chris.
There are. And they are small, of course, but there are some larger burrowing types of animals that live under the sediment. Maybe not so much here because probably isn't really deep sediment here. But certainly microbes, very small crustaceans, some worms, stuff like that would be in the sediment.
Oopsies.
Okay, we have About 3 viewers asking, can you tell us about why corals are different colors?
I don't know.
We have a lot of new viewers.
You know what? Yeah, somebody else has got to answer that one. I think I've answered
All
that
right, I've got
question
it,
about
I've got
5 times.
Trevor's got it.
Okay. The corals are different colors because of the way that they are.
That's pretty good.
Corals are different colors because
It's special.
because of the biology they're made up of, and it's arbitrary, and there's no advantage to either one.
But it brings us joy.
Because
brings
it's
us joy.
very dark.
Yeah, there's not enough light in here for it to matter. So it's just a flip of the coin or a roll of the dice or whatever you want to call it, and then that species, it picks that color, and now it always is that color.
That about sums it up, I think.
That's pretty good.
Yeah, it's okay.
Nice.
That's
Yeah.
okay.
Front row's taking over the biology.
You go, front row.
Watch out.
Yeah, whatever chemicals are available there to make up the cells of the corals.
That's right. Chemicals was in there too.
Oh, yeah.
Good job.
Chemistry.
Look at this
And
elite sponge.
if you want to come join the front row, offer to you.
And the first grade scientists say thank you very much from Armin Jar Elementary in Bremerton, Washington.
Yay, go
Hey.
Washington.
We are so excited. Oh my gosh, can you imagine all these little first graders?
Yeah,
Is that
In
the class
fourth
we talked to
graders?
yesterday?
yeah.
I'm not sure.
You know, there's a bunch of us who live in Washington State, and Bremerton is just a little bit south of where I live.
There he
So
goes.
Chris can bring you some of his
Little
sponge
dive bombers.
samples. You can
Oh
actually
yeah, they
feel
can
them.
go and
It's so cute. Look at it.
I'm sure they can take a field trip to your lab at home.
my little tiny lab, I don't think so.
Chris's
They can
basement,
take turns.
That might be a bit odd.
no social distancing.
Okay, where am I going? Other than just
We're just going to
amusing
coat
myself.
sponges in Chris's
I
basement.
forgot to move the ship. I was so entranced by the
We
little
can just
sea-
stay here and torture Riddles.
They're laughing up front because of the bailing of the brittle stars, which is this weird behavioral phenomenon that we've seen over and over
Bridge
again.
nav 20 meters, 130.
And the front row wanted to pester them and stay there to see how many would bail, I think.
And
Oh, it's all for
Trevor
science.
Alfonso Garcia Ariola says hello. You sailed together in the Gulf of California.
Who is this? Sorry, I missed the name.
Alfonso Garcia Ariola.
Oh, yeah.
I think he was a science communication fellow
Yeah,
too.
I think I remember him giving me a little, was it a pin or a sticker
Do you want
of
to
a
zoom?
rose? Am I remembering that right? You can write it.
Oh, yeah, because he's from Portland. Yeah,
Yeah.
Rose City. Yeah, he gave us some stuff too. He was on one of the mapping cruises.
Antonella, do you want to zoom?
Oh, hello.
Sure,
Yeah.
yeah.
So we've got, this shows you how important these sponges are to so many animals. This is kind of typical. This is a different variety of leaf sponge. I think we've seen it a couple times, this cruise, but
Thank you.
it's got
Just have a
crinoids
slight
and
heading
brittle
change to
stars.
130.
Okay.
Just got a lot of stuff on it.
Another viewer
bearing
says
change.
simply gorgia.
Yeah, it really
Simply
is.
gorgia. I like that.
I always use the wrong term.
Yeah, those boulders, big rocks with the beautiful arrangements.
Yeah, now we're getting into
Simply
some good
gorgeous.
biology or geology here. So the last watch may have a really, really nice 2
Oh
hours.
my gosh.
I think
So we have 755 viewers on YouTube now. It keeps going up.
yeah.
Nice.
Well, I'm glad we're,
Viewers.
Oh, yeah.
Why don't I come up, too? This is
That'd be nice.
So,
Thank
It's
you.
because of the snowman.
better
and
to come
I'm not
up.
sure how many classrooms are watching.
Oh,
And each
Oh, those are
classroom
bigger
has
than I
at least
thought.
20 students.
these are big.
Lots of viewers.
These are big polyopogon sponges. So I'm delighted that we have so many viewers that are able to see some of these places the last few days. This has been really pretty. And the rocks are fine, but it's kind of nice when we can also see some of the deep sea life. Well, it's a really nice combination dive here of
135?
geology.
It was 130.
oh, okay.
And then
So-
after this, if it keeps rocky like this, they'll probably just go directly east.
Okay, awesome.
It's a beautiful red stalked sea lily, or sea lily that is, stalked crinoid. We've been seeing those throughout the dive today.
Yeah.
And And a viewer says, so I think the true question is, does Chris enjoy sponge baths?
Let's see.
Sorry, I had to say it.
It's all you, Chris. We're waiting.
Okay.
So I want to know, does OET policy require me to answer every question? That was a good one, viewer. I appreciate that. I don't mind jokes pointed directly at me as long as they're good. That was funny. Thank you.
You get a lot of compliments.
And I refuse to answer that.
Oh, but you can answer this one. Any octopus sightings yet?
No, actually.
Not,
That's a good
not on
question.
this cruise, right?
We didn't get one single
No.
octopus.
I know.
Which is really unfortunate because they're really weird octopuses down here, octopi, whatever you want to call them, because they got fins on them, unlike your normal shallow octopuses. So,
Yeah.
Well, there's also Casper, but he's often down deeper.
yeah, Casper, you know, and they call these serrate octopi that because of these fins, but we didn't see one. We almost always see at least one on
We
a
had plenty
cruise.
squid, that pyre.
Yeah, we saw squid.
But none of the like cool squid down deep.
No. The,
Vampire squid.
yeah, the cephalopods, which both squid and octopi are members of, they've been
Yep.
kind of a
noticeably
no-show.
absent. Yeah, it's true.
This one. I'm sorry we couldn't have treated the viewers to some of those because they're really cool.
Oh, go for it.
Another viewer says, these dives are my new favorite pastime. It's fun, beautiful, and important work.
Yay.
Thank you
Very
very much.
extremely
Thank you very
important.
much.
Yeah, so if you want to
Oh,
want to
yeah.
provide comment on how to protect this, most of these dives have been happening within Papahanaumokuakea Marine National Monument, and NOAA is considering that area as a National Marine Sanctuary to enhance the
I don't know why
protections
I
for that
can't hear
area. So if you want to
It's weird.
provide comment on that, we're accepting
great.
comments through the end of January, and you can go to papahanaumokuakea.gov.
Yes,
and
the
we'd love to hear
navigators
from
feed
you. We're going to have also 4 public meetings in this month, in December. And those are open to the public, and you can also go to that site and look up the sanctuary designation, should right on the front page. And you can participate in the meetings and provide comment, or provide comment through the website. And please, we'd love to hear from you through the end of January. The website again is www.papahanaumokuakea.gov. Yeah, so this dive is in international waters. We are outside the exclusive economic zone of the United States, and we are in what's called the high seas, or international waters. And there are no additional regulations or protections for this area as it stands. I know that the United Nations Environmental and Scientific Organization and the UN are working on some kind of framework for high seas and these international waters and any protections they might need. But it is outside the EEZ. So it's good to see that they seem to be doing okay. We haven't seen, it's hard to determine whether there had been trawling impact or something, but I mean, Chris, you would probably know better than I if you've seen anything here that gives you an indication of that.
I haven't seen any indication of that. This is
good.
pretty deep for
Yeah.
doing either tangle nets or dredging, thank goodness.
And there's not much to fish for.
No, there isn't at these depths. So.
Yeah.
And we have another question here.
Uh-huh.
Do corals or sponges have special organic compounds in their body
Oh,
to withstand enormous pressures deep in the ocean? Compounds such as trimethylamine N oxide.
I don't know those chemicals. I'm afraid I'm not a chemicals,
Trimethylamino
but
oxide. I think
they certainly have got some special type of compounds, and they have special enzymes in there to withstand the pressure. They're called refer to them as pressure sensitive enzymes, and fish probably have them down here too. But the thing is, with regard to pressure, none of these animals have any type of airspace in them, so therefore pressure is less of a problem than it is for, say, fish, which might have an air-filled swim bladder. Even the arthropods generally don't seem to have any type of air anywhere in their bodies, so pressure, except for the enzyme activity and things, pressure really isn't that much of an issue. They're not going to have to deal with barotrauma, for example, which is usually referring to the expansion of air when an animal is brought up from the surface quickly, you know, or yeah, I guess I'll leave it at that.
Yes, thank you, Chris.
So we are due north of the summit of the seamount, I believe. So, Aaron, do we really make 2.7 kilometers on today's dive so far? Is that possible?
Is it possible? Let's have a look.
Might even be likely.
I think the end point I put at 2.7 kilometers away from the start point.
Where did they leave us?
We're coming up to the top.
Oh, you want to know from like total?
Yeah,
not
total.
just our part. Okay,
right,
yeah, that's
just total.
easy. I could do that.
And we have another important question from Miss Elliott's class
Bridge
from
nav, another 20 meters, 090.
Mountain View in Shelton, Washington.
Fish. Looks like a duckbill.
So
No.
Let's get that
we'll
weird
answer
face.
that question in a minute. We're looking at a fish. What kind of fish is this?
This is another synaptobranchiid eel.
An eel.
Yeah, these guys are pretty common on these rocky habitats.
Yeah, 2.5 kilometers straight line distance.
Wow.
Which is pretty close to what we've been doing.
Yeah, that's pretty good.
Yeah, we were hauling.
And there's could we take a look at that star too if we have a chance? I think he's headed toward the corals.
Bridge nav, hold position.
Yeah, just a brief snap zoom would be fine.
Yeah, we're pretty far ahead anyway, so it's probably better to slow down a bit.
For a watch change.
Yeah.
So this is a type of sea star that predates on corals, and I think he's looking for his next victim.
He is. I can see it in his eye.
Yeah, and he's pretty close to this marvelous tree, and he may have actually cleaned off the tree on the right, and that's why it looks all white and without any tissue on it. And I think he's
like
maybe trying to figure. Yeah,
horror story.
Yeah, it's he may be just settling out a bit and digesting and getting ready to launch himself onto this next one.
He said it could take him months and months to eat one of those coral strands, huh? Oh yeah, look at it. He's sniffing. You see that?
Yes.
He's got his little arm up, sniffing the water for the next meal.
Yeah,
Little sniffy arm.
Huh? Look. Oh, wow. It's like being laid under siege.
could be evoplosoma or something like that.
Pictures. Takes pictures of that guy. Death.
It's a beautiful
Stalker.
bamboo coral. There might be a little white sea star right on the, or is that barnacles maybe? I don't know if we can do a snap zoom. Oh, I think they're barnacles. But let's take a look.
Can you zoom in, please, Aaron?
Yep, can do.
A little cluster of barnacles. Procylosmatidae, if I remember the name correctly. For the family of these little coral-dwelling gooseneck barnacles. There's a basket star that's over to the right. They're pretty common. These are the types of basket. There's a brittle star to the left. So you have a basket on the right and brittles on the left. They superficially, they look almost the same. And
Scott
what
saying
is, oh, that's
iClade.
that's the sea star.
Oh, wow. Take a picture of that. To like
This
that.
is an
Stalking
I-clade. This
hunter.
might be J-clade. It's also a nodal branching.
Scott says iClade.
Scott said I-clade? Really?
I don't know. That's
Oh,
what I typed.
okay. So in the Isodella group, okay. Oh. Huge tree, look at that. Well, good sized tree. We've got a sparse one over on the left. I don't know if it's possible to get over there to the left.
Antonella has to come back out in front of Argus.
Okay. Yeah. Okay. But just to give everybody a heads up, we're going to be relieved in probably about 5 more minutes with the next watch. And then I believe they're going to have about somewhere between 2 to 3 hours before they've got to come up, and we're going to have to say goodbye to this wonderful group of seamounts and start heading to Honolulu.
Yeah, it's been quite a pleasure.
Yeah, so the question is from Miss Elliott's class. Have you found any new species during this expedition?
Well, until we get a chance to examine them back in the lab, we can only call them potential new species, but the answer to that is yes, absolutely.
Mm-hmm.
So, and right now I can't sort of rattle them off, I don't think, because everything's all jumbled up in my brain, but I think we've got some new species of sponges. I think we potentially have a new species of crinoid. We have some potential new species of what they call a polyplacophorine, which is a type of mollusk, sort of a slug type thing. I'll leave it at that, but yeah, I think we do.
And many microbes, new species of microbes.
Yeah,
possibly.
that's right. Excuse me. Don't forget the
In
microbes. In fact,
Yes.
there may be more
Yonder.
species of
I'm
microbes
going
than there
yonder.
are of any of the big ones.
Yeah.
Yeah.
Species of rocks. Just
Yeah.
kidding.
Doing a great job.
So thank you to all of our viewers for following us on nautiluslive.org. We're just about to end our 4 to 8 watch here, Hawaiian Standard Time. There is another watch coming, and our dive is expected to end at noon today, right?
That's correct, but we'll be at the surface at noon, which means we're probably going to be leaving bottom somewhere between 10 and 10:30 in the morning Hawaii time.
So we do have another 2 hours of observations and exploration on the ocean floor.
And then some blue water for the next watch to answer questions. And that'll be Dr. Beth Orcott and Steve Oskovitz. And they've got lots of stories to tell too.
Yes. So keep those questions coming and comments. Thank you very much for following us. It has been a pleasure.
Aloha.
Yeah, thank you very much to everybody. And
Bye, thank you.
Hui hua kai, over and out.
over and out. Bye-bye.
Yes, thank you.
Check.
Good morning, Team Ham.
Is this what we decided on? Team
That's what I decided.
I
Anybody
like
can call it whatever you want.
You can call it whatever you want. It just has to fit within the acronym HAM.
Yeah.
And you can include silent letters. So actually,
well, we have 110 minutes to figure out what those letters stand for.
We're gonna wait just a few moments to get started. Our science communication fellow is finishing up a ship-to-shore interaction. We want to wait for her to observe our protocol. In the meantime, Rennie, can you tell me what the latest has been with the ship moves and recommendations? Oh, you're not on SPL.
Thank you. We are holding position currently. It looks like they have kind of come on the north side of the summit here. Talks of whatever you like, but there were talks of following the contour and then heading up to the summit. We have about 2 hours on the bottom, and then we'll have approximately 2 hours of ascent and recovery.
Yeah, okay.
The winds have been picking up in just under and around 20 knots. So we'll keep monitoring that. But as of now, and it's certainly choppier out there, but as of now, it looks like we're still okay for the noon recovery.
Okay.
Yeah.
That sounds good. And Steve, what's our situation with sampling capacity?
we have a few boxes open, 2 on the starboard side, and the forward box is wide open.
Okay.
One Niskin, we're going to remain empty until we do our off bottom.
Okay, and how about the slurp chambers?
Most open,
Most
2
open.
used, yeah.
Okay, great. Yeah, so maybe, Rennie, if we can go ahead and put in a ship's move kind of towards the east to follow this contour, and we'll go ahead and start moving while we wait for our science communication fellow to join.
Can do.
And then we'll observe protocol once she's here.
Sure.
While we're, can we zoom while we're sitting here?
Sure. Yeah, I wasn't sure if they had already looked around here, but I have no...
On the upper right, there's a purple thing.
Yeah, let's go look at the purple thing.
Some of our scientists ashore have noted.
Okay. I'm still getting the science chat logged in, so... This is the final watch of the Luo'ea Ahiki Ikapapaku expedition. Thanks for everybody that's been joining us online as we've been exploring together these ancient volcanoes located within and just outside the Papahanaumokuakea Marine National Monument.
Roger.
Victor Gorgia, perhaps?
Oh, we wanted to wait for Lindsay.
Go ahead and push that in there.
Yeah, yeah.
Yep. It was a bit of a optical illusion, this one. It looked like it could have been encrusting on another coral, but it looks to be a colony of Victor Gorgia.
It's a beautiful way to start our shift.
Yep.
Come on, science chat.
All right.
Great, thanks.
Full wide there, please, Jeff.
So for those joining us and who have been regular listeners to the 8 to 12 watch, you know that we have been constantly discussing what a team name might be, shift name. A suggestion is HAM, H-A-M, the acronym, which could stand for many things, and we still welcome your suggestions on what that could stand for. We've got one in the science chat coming for have any manganese. I like it.
Highly altered mighty microbes.
Highly altered mighty microbes. Yeah, all right.
It's like a failed got milk campaign.
A bit of sediment here. Is this stirred up from the thrusters? Is this?
This is probably coming off of our vehicle. You can kind of see it behind us there. So I think it's stirred up from...
Team HAM is becoming complete. Our science communication fellow is joining us.
Good morning, team.
Good morning.
Good morning.
We're waiting to observe protocol till you joined us.
Oh, thank you for waiting. Sorry to keep you waiting.
It's okay. Shore interactions are important.
It was really fun to see stuff behind me, and everybody was asking great questions.
Great.
Well, shall we do it?
Yes, please.
Let's do it.
All right. Well, we're the final time on our watch and the final time on this expedition, we're going to be going radio silent to conduct a private ceremonial protocol that acknowledges and honors the biocultural resources that are present and that we'll be interacting with in the final hours of this dive. So we'll be back with you shortly. And at this time, I will invite everyone in the van to go ahead and mute your microphone.
All right. Thank you for taking that moment to center what we're doing here. Remember that it's a really great privilege to get a chance to see this part of the seafloor and be in this part of the world. And I'm so thankful that we get a chance to stream this live so that people around the world can be part of this exploration with us. Especially our audiences that are here in the region, in Hawaii and other Pacific Island states. Aloha, thank you for being with us.
Yeah, aloha. Looks like we do have a number of viewers tuning in from Hawaii, as well as other parts of North America, Canada, US, and South America.
Yeah, and we have a number of interactions today with different audiences around Hawaii, as well as we've had for several days now. So Kalamehu and others have been engaged in many interactions, and we have some more scheduled today. If you haven't yet had a chance to sign up, but you're interested, there are other chances for upcoming expeditions, and you can find those and sign up or request an interaction. And it doesn't just have to be for, school groups and classrooms. It can also be for other learning communities, other interest groups. You can find those if you go to the nautiluslive.org website under the education tab and look for the ship-to-shore interactions. And there's a form you can fill out there. Lindsay, just to fill you in on our plan, we're kind of following around this contour around the summit here, and then we'll probably head up towards the summit in the near future.
Sounds great. And just for viewers out there, if you've been watching for a while, you've probably been hearing this a lot, but we are experiencing some technical difficulties with our comment chat feature. So if you're using that, sending a message to our team, thanks for your patience as we intermittently get those in bulk.
All right. Steve, do you know if they ever found some really high density communities on this feature after our watch last night?
Yeah, in typical fashion, as soon as we left, things got great.
Okay.
No, but they didn't get really great until about the 4 a.m. watch.
Oh, okay.
So my understanding was that it was pretty sparse, similar to what we'd seen for the duration of the 12 to 4. And then, yeah, it sounds like the 4 to 8 watch had a pretty good time working through some of these high-density coral communities.
Okay.
And then following these contours. Much different diversity than what we had seen down deeper.
Oh, okay. Interesting.
You know, we've seen, yeah, we've really kind of swapped a lot of the bamboo corals for, we had primarily black corals, bathypathes were the most abundant. Now it seems to be these bamboo corals, a combination of iclade, a couple of different iclade species of bamboo corals, and then Acanella species here.
Okay.
Sponge-wise, we're still seeing some of those large polyopogon sponges, yeah, but also some other glass sponges.
Okay. Video, is it possible to change the gain? It looks a little dim on the sides, but maybe that's the lighting configuration for today's dive.
That's the lighting configuration.
Yeah, okay.
Yeah.
What's that white puff back there?
Looks like a sponge.
Pretty safe money.
Also kind of looks like a shower cap.
Yeah.
That's where it went.
I think that's what Chris was calling Atlanticella earlier. Sponge.
Okay.
Yeah.
Steve, can you see in the notes?
Could do a snap zoom there, please.
What kind of rocks?
That's good.
Andrea was able to collect?
Yeah,
So
on their watch they got
for anything else?
subangular,
Okay.
rough textures, some angular shapes.
Does it say anything about the kind of formation it was in?
Forward.
No, it doesn't.
Okay, that's
Just
fine.
gives sizes and textures and shapes.
Yeah. Okay. Still seems that this seamount feature has a lot of these sheet-like flows.
My chat is still loading, so if anything comes through.
We've just got let's see, a couple comments from Scott about how there's trident candelabra bamboo corals, more than he's seen on any dive before. Some sparse branchers.
Yep.
This one looks really denuded.
Scott, we got a really great imagery and collection of a sparse brancher.
Yeah, right, on our last watch.
Yep, one of the few large corals we saw.
Yeah, that's residing safely in the slurp canister right now. Rennie, do you mind backing out on high pack just a little bit so I can kind of see the dive progression?
Sure.
Okay.
We started
Yeah, way out there.
way up.
Okay.
Up here.
So it's mostly been on the north side of this feature. There hasn't been at least on the most recently, there hasn't been much on the south side.
Yes, most recently it appears that way.
Okay. Yeah, so I think we'll come around, maybe come all the way around to the south and then come up.
Okay, so I called in another 90.
Yeah, 90 is great.
We'll go south.
Yep.
We'll have to keep an eye on timing.
Okay.
I don't know if we'll get to this side.
Yeah.
But we'll definitely be able to go around here. Maybe cut over.
What is our hard off bottom time?
We need to, I think Rennie said we have 2 hours.
I think, yeah, 2 hours. That'd be good. What's your weight situation?
There's still 2 plates on board, so we can pitch those. Yeah. So it'll be all good, I think.
Okay.
And my Z-bias is pretty good right now, so.
Okay, that's based on 15, so.
Go ahead and do a partial zoom.
Steve, can you remind me of the name of these beautiful red visions here?
That's Proisocrinus ruberimus.
Right.
Someone copy-pasted them. Jake, you can turn off the lasers. All right, Jeff, you want to push on in a bit more?
Yeah, who photoshops these there?
Very charismatic.
Yeah.
Thanks, Jess.
Go wide, please.
Yeah, they the ones we've collected so far, they tend to bleach. All the pigment tends to run out during the preservation process, and so they actually turn white or very pale.
Okay.
After they've been preserved. So getting these in situ images is very important.
Yeah. Given that we don't have a ton of bottom time left in the dive, I'd like to suggest that we kind of just try to cover some ground and see what we see. Maybe because it seems that we're seeing a lot of things that have been seen already.
Sure thing.
So we might not spend a lot of time getting beauty shots, and more time just seeing how the composition changes as we move around.
Sounds good.
Does that sound good, Steve?
Yeah. Then we can kind of plan a little bit of contingency time to shoot up at the top.
Yeah, exactly.
Yep.
Yeah, I'll keep an eye on that. See where we get in about 45 minutes and then head up there. So one of the few times that our 8 to 12 watch is actually on during the daytime, and we get a very different audience than we have in the wee hours.
Getting some very kind comments. I'll have to read them when we're in our blue water.
Yeah, that sounds great. Thank you.
We've also got folks who maybe aren't as familiar with our watch tuning in, asking what our name is. What team is this? This is the 8 to 12 watch. We go by many names.
We do. we have cycled through many. Do we want to try to come back through the list of what we've tried? Blue Water Warriors, Duty and Leisure,
Savage Cribbage.
Savage Cribbage, right?
Intertentacular
Sclerites.
Right. Yeah, I don't really remember that one. But we've kind of been angling towards
highly altered
microbe.
You guys have been leaning towards those.
Other, oh yeah, I didn't necessarily pick that one. But see, things that would fall into the acronym HAM. So we've gotten also the suggestion for have any manganese. There's a couple others we had last night for that acronym.
Oh, I should have kept those.
Maybe we could also be happy with aloha and mahalo.
Oh. Steve, a viewer is wondering if all polyps in coral colonies bud from a single individual polyp.
Well, I mean, they usually settle and they'll form a single polyp, most colonies. So I guess yes, I think is I think that's what they're asking. Most colonies, when they settle, yeah, you'll see young recruits will be a single polyp perhaps on a small branch, or sometimes in the case of sea pens, they will be just one polyp sticking out of the sediment. I think we saw that last night, potentially. Could have been an anemone as well, but yeah, usually it all starts with a polyp, and you grow from there.
So if you were to look at the distal ends really closely of these, would you see budding potentially?
You know, so interestingly for the bamboo corals, that there might be some diagnostic information one could glean about whether they have polyps at the end of their branches. Some of them do tend to have those kind of very small polyps at the tips of their branches. Others do not, but they don't have to. They can regenerate polyps from within the colony. But for the most part, for example, if it's a regrowth or if it's trying to repair a wound, those cases. Oftentimes, though, even in things like primnoids, we see where the polyps grow in whorls. Sometimes they can, the whorls will have different numbers of polyps. They could be in some process of creating additional polyps or splitting additional polyps for each whorl. So yeah, it happens all over the colony. But if they're talking about how colonies grow, then usually, yeah, the branch tips is where the linear growth extensions occur. But while linear growth is happening, they're also laying down additional calcium carbonate into the skeleton all over the colony, wherever there's tissue.
Well, thank you.
How are we doing on the ship's move, Rennie?
Just now finishing up another one. Gonna call in one more. Continue east.
Yeah, okay, maybe one more east and then we'll start heading a little bit more south.
Sure.
A lot of these red crinoids on this side.
Oh, chat's back. Perfect.
Jess, are the lasers for navigation and driving the ROV?
Not necessarily for our ROV. I mean, we can get a sense of scale for when we pick things up, so that's pretty useful. They're more for the scientists, so they can tell also the size of things, but it's helpful for their kind of video analysis afterwards.
For any of our regular watchers of the Nautilus live stream, if you've been joining us for several dives of this expedition, I think we visited a number of seamounts and seen a number of really special places. I'd be curious to hear from you in the chat if you have a favorite seamount location that we visited. Was it Seamount A, which we started with, further south? Ha'eo, which was our furthest to the northeast. We had the Don Quixote Gio. We had number of dives there, as well as a couple of unnamed volcanic structures around the Don Quixote seamount. We visited Euphemia, Sovereign seamount.
Can we take a look at this yellow bamboo here?
Yeah. Oh. We had two dives, one dive on Tamana and one dive on a little ridge feature to the southeast of Tamana. And now we're here at whole Ika seamount. So let us know. What did you find most fascinating in this exploration? What are we looking at, Steve?
It's a bamboo coral.
It's a very big
get
one.
a better view of it. That's a good size.
Is there a particular place you'd like the video to zoom in on?
Anywhere where we can see branching structure. So
okay.
I think maybe mid-colony to mid-high end of the colony.
Okay.
It gives a good view of polyps and branch nodes.
Were you able to get science chat to load?
Yes. Jeff, go ahead and push on in there a bit, please.
And a reminder to those who are relatively new to joining us watching Nautilus Live, and you see these red brittle stars attached to the corals, and you may be thinking
down a touch.
Thinking they're eating the coral, but they're not. They're through the flow of water to filter out particles. Looks like it's internodal.
Yeah, it looks like, yeah, just above the node in most cases. This yellow coloration is characteristic of a clade called S1, which we've seen these on other dives. We've seen pretty dense gardens of these on previous expeditions, most notably with the Okeanos Explorer. There was quite a dense garden in the Jarvis Island area of these S1s.
Are these considered octocorals?
Yes, yes, yeah. All the bamboo corals. Another one of these ophiocanthids, typically associated with these larger branching coral colonies, but it seems like wherever they
Can we get the lasers on just briefly?
Tissue loss.
Pick up and go.
Sure, coming on.
And we just saw some there.
Thank you very much.
Yeah, sure thing. It's a very, very yellow coral.
Yeah, yeah. Typically, we usually see the yellow wash starting from the base, working from the base up. Yeah, they are particularly striking. I think this is the first one I've seen, at least in a few hours. So we'll see if they seem to be more abundant at this site as we move on. But that was very, very large.
Yeah.
Colony. At least a meter tall, I would wager.
Oh, at least. Looks like there's a Chaunacops.
Oh.
We want to get a partial zoom on that.
Aren't we just the luckiest? Seems like we see all the Chaunacops on our watch.
Yeah, we've seen.
They saw a really beautiful one on the 12 to 4 watch the other day. And actually took off and started swimming.
All right, go ahead there, Jeff. I think we saw a juvenile too, didn't we? A little lavender one.
Oh, okay. Is that what that was?
They're a little translucent. Parts of the body you can see right through.
Yeah,